Good morning, afternoon, or evening, and thank you for joining today's Endo-ERN webinar entitled Rare Diabetes Caused by Endoplasmic Reticulum Stress, Clinical Features and Novel Patient Stem Cell-Based Models. I'm pleased to introduce you to today's speaker, Professor Mirjam Knop from Kliniek Universitaire de Bruxelles, Hôpital Erasme.
So my talk today is on diabetes caused by endoplasmic reticulum stress, clinical features, and novel patient stem cell-based models. And I like to start, as always, by mentioning the people who are involved in this work in our laboratory, the ULB Center for Diabetes Research in Brussels. I've highlighted the people who participate in these activities in bold, but in particular, I'd like to mention Federica Fantuzzi and Maria Litreviv, two very talented fellows who have worked on some of the projects that I'll be showing you today. I'd also like to mention Elisa DiFranco and Andrew Hattersley in Exeter and Cécile Juliet in Paris for their work on rare forms of diabetes, and Pierre Marchetti for our long-standing collaboration human islets of Langerhans, and Timo Tonkowski for the work that we have done together using human-induced pluripotent stem cell-derived beta cells to study diabetes. So why, on a talk by the European Reference Network on Rare Endocrine Diseases, are we looking at diabetes, a disease that currently affects around 8% of our population? Well, it is because there's many different types of diabetes. The two most common ones are polygenic. Type 1 diabetes is a disease caused by the destruction of pancreatic beta cells by the immune system that affects around 10 to 15% of all cases of diabetes. Type 2 diabetes is a rather poorly defined condition of defective insulin secretion, often in a context of insulin resistance. But then there are dozens of uncommon monogenic forms of diabetes where the disease is caused by a mutation in a single gene. And as a clinician scientist, I find it very important in my clinical practice to recognize these different types, not only to pursue research on disease mechanisms, but also to guide patient care and make the best treatment choices for our patients. Now, one common pathogenic mechanism in all of these different types of diabetes is beta cell dysfunction and death. In other words, you do not become diabetic if your beta cells are healthy. And a mechanism, a cellular stress response in the beta cells that has been implicated in beta cell failure in these polygenic and monogenic forms of diabetes, and most strikingly so in the monogenic forms of diabetes, I will add, is the endoplasmic reticulum stress response. And I'll illustrate that using two vignettes in the talk today. So why the endoplasmic reticulum? You may, for some of the clinicians on the call, on the conference, you may recall that this is an organelle where all secreted proteins are synthesized in this endoplasmic reticulum. So homeostasis in this organelle is crucial for highly secretory cells, such as pancreatic beta cells, that need to produce large amounts of insulin. When endoplasmic reticulum stress develops, and that stress is defined as an imbalance between the demand that is placed on the organelle and its functional capacity, you have an accumulation of unfolded or misfolded proteins in the lumen of the organelle. And this is sensed by 3 endoplasmic reticulum stress transducers that sense there is stress in the organelle and communicate to the cytoplasm and the nucleus. So under resting conditions, these 3 proteins are inactive, But when unfolded or misfolded proteins accumulate in the endoplasmic reticulum and lumen, they will be activated. And the prototype, perhaps the first disease that I should mention here, is diabetes caused by insulin mutations that lead to insulin misfolding. The example is the Akita insulin mutation, where a cysteine is replaced by a tyrosine, and where the formation of disulfide bridge the formation of disulfide bridges in the insulin molecule is impacted, making the insulin unfolded and leading to this accumulation of unfolded proteins in the lumen of the organelle. So when these ER stress transducers sense the stress, they will be activated and communicate to the cytoplasm and the nucleus. And ATF6 and IRE1, upon activation, will induce transcription through the generation of transcription factors of endoplasmic reticulum chaperones and folding enzymes. This is the ER stress response is an adaptive response where there is stress in the organelle, and the response aims to solve the problem. Here, by increasing ER chaperone and folding enzyme expression, the functional capacity of the organelle will increase and clean up the mess in the lumen. The third ER stress transducer is PERK. When activated, PERK will trans-autophosphorylate and also phosphorylate the alpha subunit of the eukaryotic translation initiation factor 2. eIF2 alpha phosphorylation leads to global attenuation of protein translation. Again, this is adaptive. It means that there will be less newly synthesized proteins entering the endoplasmic reticulum, giving the cell and the organelle a window of time to clean up the mess here. But in parallel, eIF2 alpha phosphorylation will lead to the facilitated translation of transcription factor ATF4 and the pro-apoptotic transcription factor CHOP. So this is an adaptive response, which means that when signaling is activated, it also needs to be terminated when the endoplasmic reticulum stress has ended. And this is mediated by a number of negative feedback mechanisms on the PERK pathway. The first one is GAD34, which is induced by CHOP, a non-enzymatic cofactor for protein phosphatase 1 that will target the phosphatase to dephosphorylate eIF2 alpha and thereby terminate eIF2 alpha phosphorylation. A second non-enzymatic cofactor exists called the constitutive repressor of eIF2 alpha phosphorylation, the protein CREP, that was described by David Ron and colleagues, that will also target protein phosphatase 1 to eIF2 alpha. This is a protein that is expressed also under basal conditions. And then DNHAC3 has been described to tone down PERK activity and signaling. Now, the fine-tuned regulation of signaling in this branch of the endoplasmic reticulum stress response is critical for the function and survival of beta cells. And this is something that we have learned from monogenic forms of diabetes. And the first one I'd like to discuss is the disease that was discovered by Marc Nicolino and Cécile Juillet in France. And it started with the identification of this interesting patient who was diagnosed with diabetes at age 15 years. He was very hyperglycemic, fasting glucose levels of 13.4 millimoles and a hemoglobin A1C of 13%. He was autoantibody negative, islet autoantibodies I mean, suggesting that he did not have type 1 diabetes. And he had low but detectable C-peptides, showing that there was still some degree of endogenous insulin secretion. He also had microcephaly and growth retardation. And interestingly, his sister was also growth retarded with microcephaly, and she was diagnosed with cathotic diabetes at age 28 years. Both siblings were born to non-diabetic first cousin consanguineous parents. And because of this consanguinity in the family, Cécile Julier hypothesized that they would have inherited from the parents a mutation for which they were both homozygous. And this is exactly what she found. She found that in CREP, the constitutive repressor of eIF2-alpha phosphorylation, a protein that is encoded by the gene PPP1R15B, there was a mutation of an arginine at position 658 into a cysteine, and this arginine was actually very conserved all the way down to viral species. It is situated in the protein phosphatase 1 binding site of the CREP protein. So because of this position and also using protein structure data, we hypothesized together with David Ron that this CREP mutation would impact the binding of CREP to protein phosphatase 1. And this is what we examined in this assay, where we expressed either wild-type CREP or CREP protein carrying the patient mutation, and we co-expressed it with protein phosphatase 1. So when we pulled down CREP, we saw that the mutant CREP protein bound much less protein phosphatase 1 than the wild-type protein, as you can see here in these different lanes. Now, this destabilization or impairment in the formation of the CREP PP1 complex also led to impaired eIF2-alpha dephosphorylation, and this is something that we tested together with David by an in vitro phospho-eIF2-alpha dephosphorylation assay. So the phosphorylated eIF2-alpha protein here, when exposed to wild-type CREP and protein phosphatase 1, is rapidly dephosphorylated in this assay, with the majority of the protein being dephosphorylated after 45 minutes, whereas when we have the patient's protein together with PP1, most of the phosphorylated eIF2-alpha remains phosphorylated up to 1 hour. And we confirmed in beta cells in which we knocked down CREP expression that eIF2-alpha was hyperphosphorylated. And what we found is that this is in the rat beta cell line, where we silence CREP expression using 2 small interfering RNAs that at low glucose levels, insulin secretion is increased. So let's say the wild type, the healthy cells in black columns, when exposed to high glucose, 16.7 millimolar here, will induce insulin secretion, whereas in the cells in which CREP has been silenced, there is no further increase of glucose-stimulated insulin secretion when we go from basal 1.67 millimole glucose to 16.7. We also found that those CREP silenced beta cells are more sensitive to apoptosis, both under basal conditions and following exposure to a synthetic ER stress inducer. So this patient mutation is a fine example of a monogenic form of diabetes that leads to excessive eIF2-alpha phosphorylation and downstream signaling. It is associated with microcephaly, as is Walcott-Rallison syndrome, and also MEHMO syndrome, diabetes caused by DNAJC3 mutations. but not the diabetes that Elisa DiFranco reported in 2020 that is caused by mutations in EIF2B1 that are associated with liver dysfunction. Now, to study these diseases further, I am very keen to use human beta cells. I've, since I started working my PhD, had the opportunity to work with human beta cells in human islets, and this is really, for me also as a clinician, the heart of the matter, the best model to go for. But human islets of Langerhans only very occasionally become available for research when an explanted pancreas from an organ donor cannot be used for transplantation in a type 1 diabetic patient. So there is a great need for novel human beta cell models for diabetes research. And this has actually become possible through technological developments. As Sidney Brenner is thought to have said, progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. So one of those technological developments has been the discovery by Shinya Yamanaka and colleagues that it is possible to take virtually any somatic cell type from a healthy individual or a patient with monogenic diabetes, if you like, and to reprogram those somatic cells into induced pluripotent stem cells. This was such a fantastic discovery that he, together with John Gurdon, was awarded the Nobel Prize in Physiology or Medicine in 2012 for their discovery that mature cells can be reprogrammed to pluripotency. And a second discovery has been the discovery of CRISPR-Cas9 gene editing. This allows us to take those induced pluripotent stem cells that we have obtained from a diabetic patient that I'm calling diseased stem cells, and then correct the mutation that is responsible for the disease to generate a healthy induced pluripotent stem cell that is isogenic, so genetically identical to the diseased cell, except for the mutation that has been corrected. And the reverse experiment is possible as well. You can take healthy induced pluripotent stem cells and introduce a patient mutation or a genetic variant, if you're interested in polygenic disease, into those stem cells and generate an isogenic disease-induced pluripotent stem cell. Again, this was a discovery with fantastic impact in research and medicine, and it's generated or it's led to the Nobel Prize in Chemistry being awarded to Emmanuelle Charpentier and Jennifer Doudna in October last year. Now, the third discovery that has been very important for our research is, and this has been the work of many groups, that those induced pluripotent stem cells can then be guided to follow the beta cell differentiation in, and so generate in a dish beta cells that can either come from a healthy individual or from a patient with a rare monogenic form of diabetes. I've mentioned that human islets occasionally become available for research. Most often those are human islets from people with no diabetes, so non-diabetic individuals. Obviously, since they're used for transplantation purposes, it seldomly happens that we can get access to human islets from people with type 2 or even type 1 diabetes, but obviously human islets from individuals with rare forms of diabetes never become available for research. So this, these discoveries in the past 15 years have really changed the way we can do research. And so this is the differentiation protocol. We start from induced pluripotent stem cells and guide them through the embryologic development of the pancreas and the endocrine pancreas, first into definitive endoderm, primitive gut tube, primitive foregut, pancreatic progenitor, and then on to maturing beta cells. So the cultures are illustrated in the pictures in the bottom. This is an induced pluripotent stem cell colony that is positive for the pluripotency marker OCT4. When we go to stage 1, the definitive endoderm, the cells have lost that pluripotency marker here, and they acquire expression of the definitive endoderm markers SOC17. We continue to generate pancreatic progenitors at stage 4 with cells that are double positive for PDX1 and NKX6.1. And at that stage, we detach the cells from the monolayer culture, put them in suspension to generate in 3D small aggregates that look like islets. And so when we continue the differentiation, we obtain cells that are, as illustrated here in the bottom right, double positive for insulin and the transcription factor NKX6.1. So this is what these small aggregates look like. They are rich in insulin positive cells, also expressed transcription factor PDX1, and depending on the cell line, we will also generate some glucagon positive cells. So in terms of proportions of cells, we do pretty well. We have an average of around 40% insulin positive cells at the end of our differentiation. That is a proportion that is comparable to what native islet isolation labs obtain, showing here data from the Edmonton laboratory that is a big human islet isolation facility in Canada. And in terms of glucagon positive cell proportions, we're also in the same range. So we generate beta cells, but how functional are they? Well, they are functionally immature, and this is shown here. We do get some glucose-stimulated insulin secretion. It is higher when we add forskolin that pushes insulin secretion by inducing cyclic AMP, but it is far below the insulin secretion levels that we get when we do the same experiment with human islets, adult human islets from organ donors. Now, one way to improve maturation further is to take those cells that have been generated in an in vitro system and transplant them into an in vivo milieu. That is something that works for maturing cells from many different tissues, and we've also used it for the iPS-derived beta cells. So we transplant them into an immunodeficient mice and let the cells mature further. And that they functionally mature is shown on the right-hand side. So we perform intraperitoneal glucose tolerance tests in these animals. Initially, there is C-peptide, human C-peptide is detectable, but it is not much glucose stimulated, or not at all, as you can see. And over time, shown in the blue and then the red symbols, the graft acquires glucose stimulated human insulin secretion. This is when the animals have been transplanted with the human beta cells. Now, they of course still have their own beta cells. So we have a nice tool to get rid of those mouse beta cells, and that is using the chemical streptozotocin that is toxic specifically to mouse beta cells, as shown in this in vitro experiment. This drug kills mouse islets, but not human islets or the human-induced pluripotent stem cell-derived beta cells. So when we inject streptozotocin in the mice that have been transplanted with human iPS-derived beta cells, the human cells can take over and maintain glucose homeostasis. In a non-implanted mouse shown in green, streptozotocin injection will get rid of the mouse beta cells and the mouse immediately becomes hyperglycemic. In the animals that have been grafted with the human iPS-derived beta cells, the mouse beta cells are also killed, but the human beta cells maintain normal glycemia until the graft, I didn't mention this, but we graft the cells under the kidney capsule, so the graft can be removed by performing a nephrectomy, and then the animal becomes hyperglycemic. So together with my colleague Mariana Higuaío-Esteven, colleague from a neighboring university, Patrick Gillon, we performed more sophisticated functional tests in those grafted animals by doing kidney perfusion studies. And we see at the end of the transplantation experiment that those cells have indeed acquired fully mature human beta cell function. Here we're perfusing the graft with a solution that contains 0 millimole glucose, so there is little insulin secretion. Then we can elicit insulin secretion from those human beta cells by perfusing 20 millimoles glucose. We further stimulate insulin secretion by adding forskolin. When we remove it, we come back to the 20 millimoles levels of insulin secretion. Adding diazoxides that opens the potassium ATP channels, we fully abolish insulin secretion. We can stimulate it again with gliclazide, the sulfonylurea, abolish again insulin secretion using diazoxide, and then elicit maximal insulin secretion by exposing the cells or the graft to potassium chloride. So this resembles very well what we will see in an adult, fully mature human islet. So this is a model that we're now using to study these different monogenic forms of diabetes that pertain to the PERK pathway. And one of the conditions that we're looking at the moment is this MEHMO syndrome. That is a syndrome caused by very damaging frameshift mutations in EIF2S3. This is the gamma subunit of EIF2. It's an X-linked disease that comprises microcephaly, epilepsy, hypogonadism, mental retardation, and obesity. And what Daniela Gasparikova and Martina Skopkova reported in this paper when they described these 3 families with this damaging frameshift mutation is that those children also developed diabetes. It was non-autoimmune, and it developed at the age of 10 months in those 2 families and at 6 months in this child. So it is a quite severe phenotype where diabetes comes into the picture in this MEHMO syndrome. And what they showed in their paper is that this mutation in EIF2S3 leads to impairment in EIF2 protein function and increased signaling downstream of EIF2, as shown in these patient cells. I think it's fibroblasts that have higher CHOP expression compared to control cells. So this is a model where, thanks to our collaboration with Daniela Gasparikova, we've been able to obtain peripheral blood mononuclear cells from one of these individuals, one of these patients, and we're now differentiating them into beta cells. I do want to spend, for the second vignette, a little bit of time on this other disease that has been reported more recently, and that is caused by mutations in YPF5. Again, it causes diabetes associated with microcephaly. And this is a disease that was identified by Elisa DeFranco and Andrew Hattersley. Now, this disease is of interest because it points to a second mechanism, that is the critical role of ER to Golgi protein trafficking in beta cells. And the story starts with family 1 and family 2 that presented with young onset diabetes. It was diagnosed at 9 weeks in this kid and at 15 weeks in this boy. Both children also had microcephaly, quite severe microcephaly, -5 or 6 standard deviations below the norm, and both had epilepsy, also diagnosed in the neonatal period. And Elisa found, they were born to consanguineous parents, and Elisa found that they were homozygous for mutations in this gene called YPF5. She then identified 4 more patients in 3 additional families that also had missense mutations, homozygous missense mutations, again with very similar phenotypes. Diabetes developed early. The oldest girl developed diabetes when she was 20 months of age, and they also had microcephaly and epilepsy. And all these children were treated with insulin. So what is this gene YPF5? It is known, the protein is known to cycle between the endoplasmic reticulum and the Golgi and to regulate vesicle trafficking. And the homozygous knockout in the mouse is not viable. So as you can see, the mutations lie in different regions of the protein. All of these amino acids are conserved up to yeast, and they were predicted to be damaging by in silico softwares. So when Elisa and Andrew informed us of this new discovery, we asked, we looked into YPF5 expression. The first thing we did was to look at its expression across a range of human tissues, and we found that it's ubiquitously expressed and quite abundant in human islets or in a human beta cell line, in human pancreas, and it's also present in brain. So in collaboration with Pierre van der Hagen, we looked whether it's expressed in developing brain. This is human fetal brain at age 12 gestational weeks, where Pierre and his co-workers showed that YPF5 is present both in the ventricular zone down here, that is the area where the neuronal progenitors lie, and also in the intermediate zone and the cortical plates, that is the neuronal compartment in the fetal brain. And they noticed that it was also expressed, and this is quite unusual, in the choroid plexus. So these data suggest that YPF5 may play a role during the development of the cerebral cortex, and the choroid plexus expression suggests that it's perhaps involved in the secretion of important morphogens and growth factors during fetal brain development. So we then asked whether loss of YPF5 expression or function would have an impact on pancreatic beta cells. And we used here the human beta cell line. These were the first experiments that we did, the human beta cell line, Endo-C beta H1, where we silenced YPF5 expression, again, using 2 different siRNAs. And we found that the knockdown of this protein markedly sensitizes the cells to tapsigargin, a synthetic inducer of endoplasmic reticulum stress. In those YPF5 knockdown cells, there was more expression of CHOP, this pro-apoptotic transcription factor, and we demonstrated that this increased endoplasmic reticulum stress mediates the apoptosis that we found by performing a double knockdown of YPF5 in red and CHOP alone in green, and the double knockdown is shown in blue. So we saw protection from apoptosis induced by tapsigargin in YPF5 depleted cells. But as I mentioned, and I mentioned this for a good reason, we were of course interested in obtaining PBMCs, peripheral blood mononuclear cells, from one of the families, and thanks to the great help of Melek Yildiz, we were able to obtain PBMCs from these two young women, followed in the University Hospital in Istanbul. We reprogrammed those PBMCs into induced pluripotent stem cells and then differentiated them into pancreatic beta cells. And this is work that was done by many fellows in the lab, but it was really driven by Federica Fantuzzi. We observed that there was a few percent less insulin positive cells generated when we differentiated the patient cells into beta cells, a few percent more glucagon positive cells. But overall, there was no major developmental defect in those patient cells. So the patient cells are all shown in blue. I should clarify this. We obtained 4 iPS cell lines from those two young women, and we used as controls either cells from healthy individuals or CRISPR-Cas corrected patient cells in which the mutation had been edited and replaced by a normal amino acid. And this was work that was done in Timo Tonkovsky's lab. So the cells developed fairly normally into beta cells. We then asked whether they were functionally impaired, and in vitro that did not seem to be the case. Their insulin content was comparable between patient cells and control lines, and the insulin secretion, shown on the right-hand side, was also fairly similar between the control cells and the patient lines. We did find, in keeping with the results that we obtained in the clonal human beta cells, that these cells were more sensitive to this ER stress inducer tapsigargin, so that they were more likely to undergo apoptosis. Now, Timo Tonkovsky also generated a YPF5 knockout, so completely ablating YPF5 protein. I should say that in the cells from the patients, the mutation is a missense, so there is an amino acid replacement that is predicted to be damaging, but it is likely that that protein is still present and has some residual functional activity. So Timo generated in an embryonic stem cell line a complete knockout and showed that that cell produces less insulin and has an accumulation of proinsulin in the endoplasmic reticulum. When he looked at the cells carrying the patient mutation, that phenotype was much less evident. So the full knockout leads to more severe phenotype. And he also saw in the knockout in particular, there was marked upregulation of this ER stress marker BIP, which again was much less evident in the cells carrying the patient mutation. Another beautiful example of the demonstration of ER stress is this ultrastructural study, where in the knockout, again, the endoplasmic reticulum appeared distended and filled with amorphous material. This was something that was seen in beta cells, but not in alpha cells. So YPF5 seems to be critical for beta cell ER homeostasis, but not for alpha cell ER function. And again, in the patient mutation carrying cells, only occasionally was some distension of endoplasmic reticulum seen in beta cells that was much less evident than in the complete knockout. Timo and his team also transplanted those YIPF5 knockout and mutant cells into animals. And as I've shown you in our data, they also, when they use wild-type cells, see functional maturation with an increase in C-peptide levels over the course of several months. The knockout had barely detectable C-peptide levels, and that did not change over time. And the patient mutation cells initially seemed to produce insulin and C-peptide, but progressively failed and certainly did not mature, as is the case for the wild-type cells. So YPF5 points to this critical role of ER to Golgi protein trafficking in beta cells. And interestingly, another monogenic form of diabetes was described last year caused by mutations in TANGO1, a protein that is involved in a bulky protein exit from the endoplasmic reticulum. And then there's other diseases I've already mentioned, the insulin Akita mutation, there is Wolfram syndrome, IRE3-IP1 mutations that also cause diabetes and microcephaly, and mutations in MAMF that have been described earlier this year by Andrew Hattersley and Timo Tonkowski. So in sum, 11 monogenic forms of diabetes are caused by increased sensitivity to ER stress or dysregulated ER stress signaling. So these are very rare diseases, but there is a clustering, a high density of monogenic diabetes in this beta cell stress response. 5 of those pertain to the PERK branch, highlighting the exquisite sensitivity of beta cells to dysregulated eIF2 alpha phosphorylation and mRNA translation into protein. Homozygous mutations in YPF5 cause a novel syndrome of early onset diabetes associated with microcephaly and epilepsy. And we've shown that YPF5 deficiency sensitizes human beta cells to ER stress-induced dysfunction and death by disrupting ER to Golgi trafficking. So overall, I think that this loss of gene function in humans can be used as an experiment of nature. And these monogenic forms of diabetes provide us important insight into pathways that are crucial to develop and maintain human beta cell function. And last, the human iPS-derived beta cells provide an exciting species, disease, and even patient-relevant model to study pathogenic mechanisms and test therapies, both in monogenic diabetes, as I've shown you today, but also for the study of polygenic type 1 and type 2 diabetes. Now, before taking any questions, I would like to thank the clinical research study participants and their relatives, without whom none of this would be possible, the funding agencies, and thank you very much for your interest and attention. We still have postdoctoral positions available, so if you're interested, please don't hesitate and send me an email, and I'll be happy to take questions now. It's a question from Willem Staals.
Yes.
He says it's interesting to see how many of these rare forms of diabetes are associated with microcephaly. Is there an explanation for the association, specific subtypes of microcephaly gyration pattern? Willem, that's a question that I really have in my head for a very long time, and I think our work together with Pierre van der Hagen will hopefully help to address that question. You know, these cell types have distinct embryonic origin, but they do share a number of similarities, both in terms of transcription factors that regulate their gene expression, their electrophysiological characteristics. Both beta cells and neurons are excitable, and they have this particularity that they are both very much affected by these mutations that sit either in the PERK branch or in the pathway that regulates ER to Golgi trafficking. Why there is this sensitivity of those two cell types is not known at this moment, and I think it's a fascinating question, something that we should really work on. So here's a question from Desio. The YPF5 mutation will also lead to gene deletion in alpha cells, who are also highly secretory cells. Do you have a hypothesis why beta cells are much more affected than alpha cells? It's again an excellent question. It shows that, you know, beta cells and neurons are more similar than beta cells and alpha cells that are embryologically not so distinct. The short answer is we do not know. It seems that cells that have dysfunctional or reduced YPF5 expression that need to transport insulin from the ER to the Golgi are affected, whereas cells that express glucagon have no problem whatsoever. We haven't actually looked at alpha cell function in those cells as yet. That is something that we can still do in the models that we have. But there is really no clear answer for that. What comforts the sensitivity of beta cells is the description of the Tango1 family. It was a description of a family in Ghent. We have also obtained PBMCs from those patients. It's a single family that has been described so far that has diabetes with an onset in childhood or adolescence. All 4 siblings in that family are insulin treated. It's associated with intellectual disability, not with microcephaly, at least it's not been described, let me put it this way. Endocrinologists are not great in measuring the head circumference of their patients, so I wouldn't exclude that there is no microcephaly. And they also have, and this was the phenotype that actually brought the clinicians to go looking for a mutation, they also have dentinogenesis imperfecta, so they have a quite important dental phenotype. And so Tango1 has been described to be involved in the exit of bulky cargo collagen, you know, which is probably responsible for the dental presentation. So bulky cargo exit from the endoplasmic reticulum, but insulin is not bulky cargo. So there must be something that makes beta cells particularly sensitive to these alterations. I hope I'm answering the questions. And then we have Donnelyn Schoner. I don't see her question as yet. Michael Cowett says, can you speculate on what you think might be the mechanism of YPF5 in beta cells via protein-protein interactions? That's an excellent question. We're not sure. I have heard of YPF5 interacting with another protein that's involved in ER stress-related diabetes, but it's not my data, and I'd rather not mention it in public. So something that needs to be looked at for sure. Thank you for that question. And then there is Donnelly Schooner. You mentioned, hi Donnelly, you mentioned that understanding these ER stress monogenic forms better can lead to patient treatment strategy. Can you cite an example you have in mind at this point in time? Well, we haven't got any data as yet, but we have in the past found that GLP-1 receptor agonists protect beta cells from endoplasmic reticulum stress induced by free fatty acids by modulating the way the beta cell responds to ER stress and also increasing its anti-apoptotic defense. So I think that may be one approach to test. And again, the patient iPS-derived beta cells are a fantastic model to do that in a dish. And even in the humanized mice, we could do those tests of different therapies. So Farouk Syed, hello, asks, do any of these mutations found in type 1 diabetes populations? The short answer is no. There is, as far as I know, no description of genetic variation in these particular genes in patients with type 1 diabetes. But what I can add is that, and this is work that's been done by Decio and his team, is that there is signaling in the PERK, IRE1 and ATF6 branches in conditions that are relevant for type 1 diabetes. For example, when pancreatic beta cells are exposed to pro-inflammatory cytokines, you will see endoplasmic reticulum stress. So it may be that in type 1 diabetes, and the same is true for type 2, you have activation of this endoplasmic reticulum stress response, not due to genetic perturbations, but because of environmental insults that occur in the two diseases, exposure to inflammatory cytokines for type 1, exposure to saturated free fatty acids in type 2 diabetes, for example, and that you generate beta cell dysfunction and perhaps even death as a consequence of this stress response. And then a question from Professor Pietro Maffei. Do you think we consider different treatments for diabetes in relation to the specific mutations? So for the types of diabetes that I have discussed today, we do not as yet have data. I can mention that my colleague Mariana Higuayo Esteve has looked at the impact of GLP-1 receptor agonists in the context of Wolfram syndrome, and again using induced pluripotent stem cells, both in vitro and in humanized mice. She sees that there is a beneficial effect on beta cell function and survival. So at least in that form of diabetes that is also related to endoplasmic reticulum stress, those GLP-1 analogs seem to be beneficial. We have, I will call it anecdotal evidence in patients with Wolfram syndrome that were on insulin pumps, that the addition of a GLP-1 analog has enabled them to reduce insulin doses and even discontinue insulin therapy in some of the patients, but a formal clinical study has not been done. So I think I would be the last person to advise all Wolfram syndrome patients to stop their insulin therapy and switch to GLP-1 analogs. I'm quite conservative as a clinician. Now, of course, in other rare forms of diabetes, etiology-specific or gene mutation-specific treatments exist. I think the beautiful examples are the use of sulfonylurea in patients with HNF1 alpha mutations or HNF4 alpha, or in patients with neonatal diabetes caused by KCNJ11 and ABCC8 mutations that impact the potassium ATP channel. So there is already in the field of monogenic diabetes, genotype-specific therapies available. Thank you for all the attendees for participating and asking questions.